Okay, hello tout le monde. Hello everybody. Right, so this class is going to be in English, okay? And what I'm going to do is try and give you an overview of the anti-infectious immune response. So, in my simplistic kind of vision of the world, what is going to happen if you get infected is well, the pathogen is going to uh, arrive, and every infectious disease is going to be characterized by an incubation period, which is defined as the time in between infection and the appearance of the first symptoms. So during this time, you've got a bit of amplification of the pathogen in the body, and the beginnings of the host immune response to the infection. Then you have disease, some kind of symptoms appear, and this is generally accompanied by inflammation. It's the beginning of the host immune response. And then after you've got the appearance of symptoms, you can have three potential outcomes of the infection. Either the host response will be effective, the pathogen is eliminated. That's it, that's good. Okay, so that's a win. Or you can have a highly pathogenic acute infection, something like Zaire Ebola virus, rabies, something like this. You have an acute infection, symptoms get worse and worse, and you die. Okay, and this is a win for the pathogen. Or you can have a chronic or a latent infection. So this is kind of a draw, okay? Score draw is a chronic infection, 0-0, maybe latent infection, okay? And of course, if you do nothing, then this is what's going to happen, okay? You'll either be killed or you'll have a persistent infection. So that's why anti-infectious immune responses are important. And this is really why the immune system actually exists. It exists to protect us from, being attacked and exploited by different types of pathogens. So for bacterial pathogens, we're just like a huge, massive hamburger that they want to eat. Okay? So we need to be able to resist them. Now, the first problem here is, of course, how do you even know if you are infected? So detection, pathogen sensing is extremely important. And that's the first step that needs to occur. Then after that, you can have the activation of innate, and then afterward, and then secondly, adaptive immune responses. So we're going to look at these, well, three aspects of the anti-infectious immune response. And probably I'll be talking in most detail about the first step, the pathogen sensing, because this is what is really specific about anti-infectious immunity, how to detect the presence of bacteria, fungi, viruses, because everything else depends on that, really. Okay, so if your first step fails, okay, if your first step fails, none of the rest of the immune response can actually occur. So the first step is really, really important. Now, for bacteria, fungi, and parasites, there are biochemical differences between the molecules that make up the pathogen and our own cells. And these can be detected by different types of PAMPs. Well, okay, no, the different types of pattern recognition receptor, which can detect pathogen-associated molecular patterns, which are PAMPs. So for bacteria, this will be things like LPS, peptidoglycophagelin, things that are characteristically on the outside of the bacterial cell. Same thing for fungi, beta-glucans and mannans, they're on the outside of the fungal cell wall. And in parasites as well, you have glycophosphatidyl'inositol linked glycoproteins and LPG, it's not liquid propane gas, I think it's lipoproteoglycans. So characteristic types of components of the cell wall which can be recognized by PRRs. Now for viruses, viruses are produced by our own cells. So there's no biochemical difference between protein in a viral capsid and a protein that's part of our own, of our own cells. So there's no real PRR and no real PAMP. Sorry. So what tends to be detected by the pattern recognition receptors that detect viruses is when you've got the wrong type of nucleic acid in the wrong place in the cell. Okay, so there are 5 types of pathogen recognition receptors: the C-type lectin-like receptors, the CLRs, the toll-like receptors, RIG-I-like receptors, RLR, the cGAS-STING pathway, and the AIM2-like receptors, ALR. So I'm going to take a look at each one of these, and the aim is to try and give a biochemical view of what exactly these PRRs detect. Okay, so C-type lectin-like receptors, the CLRs. Lectins are proteins that can bind to glycans, so they are going to be recognizing specific glycans. They're expressed on the surface of myeloid cells, and most of them will stimulate phagocytosis and mononuclear phagocyte activation. So, for example, you have dectin-1, which recognizes beta-glucans. So this is a specific disaccharide motif that is not present in, vertebrate cells, present only on yeast and fungi. And then for the rest of them, MINCO, dectin-2, DC-sign, the mannose receptor, often there's some kind of mannan, some kind of mannose being the key element for the recognition. Now, all of these CLRs, they've got a carbohydrate recognition domain on the extracellular side, and their signaling pathway is going to be dependent on this complex of CARD9, MALT1, and BCL10. So CARD9 is the essential signaling intermediate here. Okay, and these types of glucans or glycans, they're present on fungi and on the outer surface of bacteria. Okay, so let's have a look at this. So dectin-1, the gene name is CLEC7A, so specifically binds glucans in a beta 1 to 3 bond, and soluble glycans will not activate dectin-1. It will only work with a particular antigen. So if you've got a whole yeast cell, this will activate dectin-1, and that's going to activate CARD9, then indirectly NF-kappa B, so the phagocyte will be activated. This will induce phagocytosis and the secretion of pro-inflammatory cytokines like IL-6, IL-1 beta, IL-23, IL-12. And then what kind of immunity will you get from these cytokines in T cells? Th1 and Th17. Okay, so antibacterial, anti-mycotic immunity. Now, what happens if you don't have dectin-1? Okay, so there are some extended families which have got a recessive mutation in the dectin-1 gene. So here the carbohydrate recognition domain is interrupted by this single nucleotide polymorphism. And in this family that was identified in 2009, you can see that individuals who are their PBMC are incubated with yeast cells, and then you can look at the IL-6 secretion in the activated PBMC. So for someone with the wild type gene, you get certain amount of activation. People with the heterozygous mutation, it's lower, and with the homozygous, almost no response here. And what did these people have? Well, they had chronic recurrent vulvovaginal candidiasis in the two cases, and otherwise they had fingers and toes with a chronic mucocutaneous fungal infection. Okay, so if you lack this one CLR, you're going to get chronic fungal infections. Okay, dectin-2, similar kind of thing. This time it's a receptor for mannose glucans, and you find these in fungi and mycobacteria. And this alpha 1 to 2 linkage, you can find it in a lot of different fungi, like different species of Candida, Saccharomyces, and Malassezia, and also on the outside of some kind of bacteria, bacterial LPSO antigens, and also in Mycobacterium tuberculosis. So, it's wide spectrum of bacterial and fungal pathogens have this kind of mannose alpha 1-2 motif. Now, if you remember your biochemistry and cell biology, you will think, well, like I did, hey, look, mannose, we've got loads of mannose in glycoproteins, right? So how come this CLR doesn't get activated? Well, as our glycoproteins are matured, can't remember if this, I guess, in the Golgi, then what tends to happen is these oligomannose glycans are processed, so these alpha-2 linkages are removed, and they're replaced by sialic acid on these mannose-bearing glycoproteins. So our glycoproteins, if they're correctly processed, will not have this mannose alpha 1 to 2 linkage. Okay, so in some viral glycoproteins, you have this. So some viral glycoproteins can be detected by these CLRs that detect mannose glycans. Okay, so in terms of function, then dectin-2, once again, will activate this card9 complex, induce an FKB, and then you get IL-23, IL-1 beta. So they'll more specifically be inducing Th17 type immunity. Let's have a look at another one. So MINCO is another one of these CLRs, specifically binds Mycobacterium tuberculosis cord factor and a bunch of other mannose glucosyl glycolipids. So the sugar part is important apparently, but also you've got this acyl group here, the lipid kind of group of these glycolipids is also important. So you don't find any like glycan polymers on their own, they're always associated with some kind of lipid. And it's got a kind of dual recognition function as being a pathogen recognition receptor and also a danger receptor because it can be activated by glucosylceramide from dead cells. So once again, activation of MINCO will induce card9 on NF-kappa B activation, secretion of IL-1 beta, IL-6, IL-12, and IL-23. You get Th1 and Th17 type responses. Okay, so the key intermediate in CLR signaling is card9. So card is for caspase recruitment domain containing protein. So card, caspase recruitment domain. And once again, there are some families who have deficiency in this protein, in the gene rather. So you've got this Iranian family with a history of chronic invasive candidiasis. And if you have a look, all of the people who were infected, now look at this family. You've got kind of first cousin marriages, second cousin marriages. So you've got this consanguinity which allows these rare mutations to accumulate, to achieve some homozygosity in some people. Okay, so that's why you get these rare mutations coming up in some particular families. Okay, so if you lack card9, then you can't really resist candida, yeast infections. And if it's one of and other families of or individuals have been identified. So if there's some kind of patient with an unexplained invasive fungal infection that recurs, then card9 deficiency is one of the things that the clinicians will be looking for. Okay, so that's the CLRs, and yes.
Just a question. You said that this kind of patient with lack of card9 or like Dexin1
Yeah.
Got more sensitivity to fungal infection.
Yes.
But we saw that all these CLR also recognize bacteria.
Yeah, but you have a lot of redundancy in the system. Okay, so you're not having one receptor only for one particular type of pathogen.
Okay.
So these, Dictin-2, for example, will also be involved in sensing the presence of bacteria, but it's not the only PRR that's involved in sensing bacteria. So it's less affected. If you knock down card9, then it's really only essential. Okay, its most important role is for antifungal immunity. Okay, it's contributing to antibacterial immunity, but there are other receptors around that can compensate for the loss in that case.
So CLR is more for fungi specifically.
Yeah, I think the individuals who have these rare mutations in different CLR, their biggest immune deficit is in antifungal immunity. And that's certainly shown with the card9, because if you knock out the card9, you can't get the signaling from any of these CLRs. Okay, and the big thing they don't have mycobacteria, they don't have sepsis, bacterial sepsis, they don't have viral infections. They've got these yeast infections that are invasive. So it's not just cutaneous. They're also people are having a real difficult time in mounting an effective response to yeast antigens that get under the skin and into the mucosa, okay, or into the submucosa. Okay, so leaving CLRs, the next families that are the TLRs. So these are characterized by an extracellular domain with lots of leucine-rich repeats, so several of them. They'll be recognizing different components of bacterial cell walls for TLR 1, 2, 5, and 4. And for TLRs 3, 7, 8, and 9, they are recognizing nucleic acids. So some of them are extracellular, TLR 4, 1, 2, 6, and 5. And TLR 3, TLR 7, 8, and 9, they are endosomal. Now, all of the signaling depends on this adapter MID88, except for TLR3. So in a lot of, experiments in the mouse system, you'll see MID88 KO mice being used. So if you have some kind of effect, then that means TLRs are important, except maybe TLR3, okay? So if you knock out MID88 in a mouse, then you won't have any TLR signaling apart from TLR3. So one of the things that is, I think one beautiful thing in structural biology here is the way that these leucine-rich repeats, they form a kind of hook structure. And one way or another, the signaling is activated by the dimerization of two TLR molecules. Whatever the component or the molecule that is recognized, it's the dimerization that's induced by binding the ligand that induces signaling. And the signaling is through these TIR, toll, IL-1 receptor domains. Okay, so let's have a look at some of them in more detail. So TLR4 is very important for sensing the presence of lipopolysaccharide that, as we all know, is specifically in gram-negative bacteria. And in LPS, there are three different parts. There are the O antigens, which are very variable, the core polysaccharide, slightly less variable, and the lipid A part, which is the most conserved, identical in all LPS. So it's the lipid A part of the LPS structure which is recognized by TLR4. Now TLR4 doesn't work on its own, because if you've got a gram-negative bacterial infection and some of these gram-negative bacteria are destroyed, you'll get the LPS forming micelles. And the LPS molecule has got to be extracted from the micelle in order for it to be loaded onto TLR4. So this is the job of LPS binding protein, soluble protein, LBP, which can extract LPS from the micelle, load it up onto CD14, which can be present either as a soluble extracellular domain or on the surface of monocyte macrophages. And so the interaction here will load up one molecule of LPS onto CD14, which will then transfer it onto TLR4 and this adapter MD2. So you need this kind of chain of different molecules involved in extracting and loading the LPS onto TLR4. Oh, that's just what I said. Damn it. Okay, so looking more closely at the structure, what is specific about LPS here is you've got these 6 acyl chains attached to these two, what's that, N-acetylglucosamine by the looks of things, N-acetylglucosamine phosphate. So you have these 6 acyl chains that have to be present to induce the dimerization between the two TLR4 substitutes. So that's the molecular structure that's being recognized here. Okay, it's these 6 acyl chains attached to these two sugars. And we don't have that in any of our lipid molecules. Okay, no eukaryotes have this. So about 15 years ago, there was an effort to develop LPS antagonists. So it's a kind of lipid A analog with only 4 acyl chains. And this worked in vitro, in cell culture. You add it and it will prevent monocyte macrophage activation in response to LPS. So it was tried as a in clinical trials to try and treat patients with sepsis, to try and damp down the excessive inflammation. Unfortunately, it didn't work. Okay, so beautiful idea, but did not work in clinical practice. Okay, TLR2 is mostly going to be activated by motifs in gram-positive bacteria and some protozoa, and specifically recognizes lipopeptides and lipoproteins. So TLR2 only works as a heterodimer with TLR1 or TLR6, and the TLR1-2 dimer will recognize triacylated lipopeptides, and TLR26 recognizes the diacylated lipopeptides and lipoproteins. Now, TLR2 can also recognize the glycophosphatidylainositol linked glycoproteins from some parasites like Leishmania and trypanosomes and Plasmodium. So sleeping sickness, trypanosomiasis, malaria, and Leishmania. Now, looking at the structure here, okay, this is kind of the same as what you have in a GPI-linked protein in our own cells. So it's not entirely clear to me what the mechanism is for the specificity. Okay, we're talking. Yeah, so something a little bit unclear for me, but these GPI linked proteins from parasites can also activate TLR2. So there are synthetic antagonists that have been developed for TLR2, so PAM2 CSK4 and PAM3 CSK4. Sometimes you see these in publications. They're used as adjuvants for vaccines. Okay, so you have this sometimes in, don't know if they're used commercially yet, but they're in a lot of publications in mice. You can see PAM2 CSK4, PAM3 CSK4 as used as adjuvants. Okay, so that's TLR2, TLR4. We're going to talk about TLR5, TLR3, 7, 8, and 9. Their function is to detect nucleic acids, and they are endosomal. Okay, so they are not detecting free nucleic acids out in the cytosol, out in the plasma or the extracellular fluid. They only detect nucleic acids that are liberated in the endolysosome. And that kind of makes sense, okay? Because, you know, what you're detecting is a nucleic acid that is protected somehow from RNase and DNase 1 that are in the plasma. So nucleic acid in some kind of protein coat. Well, that's a virus, right? That's the definition of what a virus is. So if you detect nucleic acids that are only present or only released in the endolysosome, that's a pretty good way of being sure you've got a virus there. Okay, so that's really the idea here. So TLR3 recognizes double-stranded RNA, TLR7 and 8 single-stranded RNA, TLR9 DNA. Now the endosomal targeting of these TLRs depends on this protein called UNC93B. And the common feature of their activation is that they will stimulate interferon production, type 1 interferon. So if you lack some of these TLRs, what happens? You've got TLR3 or UNC93B deficiency. These are rare mutations that are associated with herpes simplex virus encephalitis. And big name here, this is from Jean-Laurent Casanova's group here. He's done a load of work on this, and one of the top people in, well, I think immunology worldwide, I guess. If you've got mutations in TLR3 and TLR7, this is associated with severe COVID-19 infections. Now, another way to detect nucleic acids in the wrong place or the wrong type of nucleic acids is detecting cytosolic nucleic acids. So this is the role of the RIG-I-like receptors, the RLRs. And there are really only two of them that detect viral RNA, that's RIG-I and MDA5. The adapter molecule is MAVS, which is associated with the mitochondrial membrane. So the signaling here will occur on the outer membrane of mitochondria or the association of mitochondria with the endoplasmic reticulum. So, the cell cytoplasm is full of RNA. So how can these molecules distinguish between viral RNA and normal cellular RNA? Okay. Well, we know this for RIG-I because it binds short double-stranded RNA molecules with a 5' triphosphate or diphosphate. Now, you might not remember this. Well, for messenger RNAs, sure, everybody knows messenger RNAs have got a 5' cap. So capped mRNAs will not bind to RIG-I. And also, the rRNAs and transfer RNAs, they don't have a triphosphate or a diphosphate, they have a 5' monophosphate. The other thing that tRNAs have, anybody know this? Something biochemically unusual about tRNAs? They've got a lot of modified nucleotides. So you often have pseudo-uridine and not uridine. Okay, and these modified nucleotides will also not activate RIG-I or MDA5. So MDA5 binds long double-stranded RNAs. Now, these RIG-I-like receptors essential for activating the immune interferon response in cells infected by RNA viruses. So the TLRs, they'll mostly be expressed by mononuclear phagocytes. So they're detecting viruses that are on the outside of the cell. Okay, so the phagocyte isn't necessarily infected. The RLRs, infected cells, any type of cell, epithelial cells, will be activated and start expressing interferon beta after activation of RIG-I-like receptors. Mostly RNA viruses, but also DNA viruses. Why? Because often in DNA virus genomes, you have the DNA transcribed in both directions. So you get overlapping RNA molecules that give you double-stranded RNA, okay, which you often don't have in cellular genes. Yes? Okay, so going over this again. So cellular RNAs, okay, messenger RNA, it's got a 5' cap. Okay, whereas RIG-I will only recognize RNA with 5' triphosphate. So this is not the same structure at the 5' end. Biochemically, it's different. And it's the same for the ribosomal and transfer RNAs. They have got a 5' monophosphate. That's different. So for RIG-I, we know, okay, why will only be activated by viral RNAs. For MDA5, it's not so clear. But you need a long double-stranded RNA. And you often get that when RNA viruses replicate, the whole genome's got to be replicated. So you've got to have a big double-stranded RNA molecule. And in DNA viruses, you often get these double-stranded RNA molecules formed. But the molecular basis, not so clear for MDA5. Yes.
You said that RR is to detect virus in infected cells.
Yes.
But when we have a endosomal TLR,
Yeah.
virus have to be in the cell also.
Okay, the question is here, TLR, it's in the endosome, the virus is in the cell. Is that, yeah, but the virus can be in the endosome. It's going to be destroyed by the proteases that are activated, going to be destroyed by the RNases and the DNAs in the endosome, which release the fragments. That cell will not necessarily become infected because the virus could be destroyed in the endolysosome, but it will activate TLR4. RIG-I-like receptors will be activated in a cell that is really infected with the virus replicating inside the cell. And similar kind of thing is the cGAS sting. So RIG-I-like receptors, mostly RNA viruses. cGAS sting is specifically for DNA viruses. So cGAS is cyclic GMP-AMP synthase, which has the ability to bind double-stranded DNA in the cytosol. After this binding, it will catalyze the production of cyclic GMP-AMP, which is a small secondary signaling molecule. So this will diffuse freely inside the cytosol and then activate the STING, which is for stimulator of interferon genes, and it's bound to the endoplasmic reticulum membrane. So once again, activation of cGAS sting will lead to induction of type 1 interferons. Now, what is cool about cGAS is that it can diffuse into neighboring cells. Okay, so this cell right here, this cell will be infected. It's got double-stranded DNA in the cytosol, which is not normal, okay, not normal for a cell. but the cyclic GMP-AMP can diffuse into a neighboring cell and activate interferon secretion in the non-infected cells. That's very cool. Now, the other thing that is great is that envelope viruses that can be released from this infected cell can also package some cyclic GMP-AMP into the virus particle. So as soon as the virus infects the next cell, the cyclic GMP-AMP can activate STING and activate the antiviral response almost immediately. So CGAS STING, interferon response in cells infected by DNA viruses. That's the main thing. Now, that's interferon. The other aspect of infection of cells is the induction of inflammation. So this is a function of the AIM2-like receptors. And AIM2 is another cytosolic receptor for double-stranded DNA. So double-stranded DNA will bind to AIM2, which will then recruit ASC through its pyrin domain. So ASC is apoptosis-associated speck-like protein containing a card. Once again, bound to the endoplasmic reticulum membrane. And this will form some kind of filament that is constructed along the double-stranded DNA molecule. So you get this nucleation of a filament, which will then polymerize, and the polymerization will recruit caspase 1. And that's what constitutes an inflammasome. So the caspase 1 will become activated. It's a protease. It will cleave different substrates that then become activated. So the big classical one is pro-IL-1 beta is cleaved to IL-1 beta in the cytosol. To be active, IL-1 beta has got to be secreted, right? But it's not in the endoplasmic reticulum, it's not in the Golgi. You can't use the classical secretion pathway. And the secretion is via these pores in the plasma membrane that are formed by gasdermin. Gasdermin is another substrate of caspase 1. So when gasdermin gets cleaved, it will be able to form these pores in the membrane, and then IL-1 beta comes out. Okay, so that's AIM2-like receptors, and so you've got this cell death because you have caspase activation inside the cell associated with inflammation. So this is called pyroptosis. Now there are 4 human AIM2-like receptors, but only AIM2 activates the inflammasome. IFI16 is another innate DNA receptor. Now, this pyroptosis inflammasome activation is also a function of the NOD-like receptors, the NLRs. So there's a big family of these NOD-like receptors that have the ability to recognize different molecular patterns associated really pretty much with bacterial infection. Okay, so if you've got bacterial infection in the cytoplasm, the NLRs are going to be activated. So DAP, that's diaminopimelic acid, so it's in the peptide crosslinks in peptidoglycan. MDP is the muramyl dipeptide, the NAM-NAG repeats that make up peptidoglycan. So 2 different NOD-like receptors can recognize this, and NLRB recognizes bacterial flagellant. So if you've got intracellular bacteria, you get activation of NOD-like receptors. So they've all got either a card domain or a pyrin domain, and that's what's going to recruit the ASC and the inflammasome. So you get formation of an inflammasome, which will give you, the same kind of things we saw before. Caspase 1 activation, cleavage of IL-1 beta, secretion by the gasdermin pores. Now, if you have gain of function mutations in NOD1 and NOD2, That's these ones up here that are recognizing peptidoglycan and the diaminopimenic acid. So these NLRs are constitutively active or more sensitive to activation. This is associated with Crohn's disease and inflammatory bowel disease. Okay, so you've got chronic hyperactivation of these receptors that's related to these chronic inflammatory diseases. Okay, so that's a very, very long section on how to detect if you are infected. And next thing that's going to happen is, one way or another, innate immune responses are going to be activated. And these innate responses are constituted of secreted proteins, complement, pentraxins, lectins, and interferons. And there's also a cellular component to the innate response. And this is going to be activated hours or days after infection. So, so what are the critical inflammatory mediators released after PRR activation? So these will be IL-6, IL-1 alpha and beta, and TNF alpha. So they all are pyretic, so they can induce fever. IL-6, IL-1 will induce the expression of the acute phase proteins in the liver, and IL-6 and TNF alpha will activate and recruit neutrophils. IL-1 and TNF alpha have an effect on endothelial cells. So these are the kind of critical inflammatory mediators in a lot of diseases. So there's been an effort to develop therapeutics to try and inhibit these mediators. So tocilizumab, sarilumab are monoclonal antibodies against IL-6, and they are approved for treating rheumatoid arthritis. Anakinra is a kind of soluble IL-1 receptor. Canakinumab, monoclonal antibody against IL-1 beta, also approved to treat rheumatoid arthritis. And these monoclonal antibodies against TNF alpha are approved to treat immune-mediated inflammatory diseases, inflammatory bowel disease, rheumatoid arthritis. Now, they've been tried in acute infections like COVID-19, but without really any very clear clinical benefit. So the application of these monoclonal antibodies or recombinant proteins that inhibit IL-6, IL-1, and TNF alpha is really in these chronic inflammatory diseases. Okay, so so these are going to be the common mediators induced by any type of anti-infectious immune response. Now, innate responses against small particulate antigens, bacteria, fungi, not so much against parasites. What's very important is complement system, which, as I'm sure you all know, after complement is activated, the three main effectors are going to be C3b, which will be deposited on the surface of the pathogen and lead to opsonization, that is, makes it easier to phagocytose. C5a is soluble. It's going to be a chemoattractant for other cells, and it's got pro-inflammatory activity. And you have the formation of the membrane attack complex, which will lead to cell lysis, particularly of gram-negative bacteria and protozoa. So how is the complement system activated? Well, there's three pathways to activate complement. Classical pathway, activated by antibodies, so that's not really innate, but also the pentraxins, like C-reactive protein, SAP, pentraxin 3. So the secretion of these molecules will be induced by inflammation, okay, either by mononuclear phagocytes or by the liver. And these are pattern recognition molecules as well. So they'll bind to phosphorylcholine, for example, on the outside of bacterial cells, and that will lead to the activation of complement. The second pathway is the lectin pathway, and here the activation of complement will be initiated by the binding of these lectins, like the mannan or the mannose binding lectin, and faculins. So the recognition of the pathogen depends on the presence of particular glycans on the surface of the cell. And finally, the last mechanism of complement activation is the alternative or the properdin pathway. So this is kind of cute. So pentraxins, lectins, you have some kind of soluble pattern recognition receptor that's going to recognize a particular molecular motif on the surface of the cell. But what if your bacterium doesn't have that? Okay, is there a way to activate complement? Yes, there will be the properdin pathway. Because what happens here is there's a kind of spontaneous turnover of complement in the plasma, and this will lead to a low level of spontaneous deposition of C3b on a biological surface. Now, our own cells have got glycoproteins or cell surface glycoproteins which will inhibit complement activation, like factor H and CD46. So this C3b will be inactivated. But if you've got C3b which is activated on a surface of a bacterium, there are none of these inhibitors. So it will carry on getting deposited and you can get full complement activation. So this is kind of like, it's not really detecting the presence of something specific to a pathogen. It's just saying this is not human. Okay, whatever I'm binding to here, it's not inactivating me, is not human, something else. So direct lysis of cells, but opsonization is very important role of complement. Now, opsonization means that some kind of phagocyte will come along and internalize the bacterium or the fungal cell and destroy it. So the first responders to a site of inflammation are neutrophils. So they can phagocytose and kill bacteria, yeast, parasites, and fungal spores. And they have activation in two steps. Firstly, chemokines will prime the neutrophil, and then a second signal like complement factor C5a and IL-8 will activate them. And these activated neutrophils, really, they kind of like explode. Okay, so there's intense production of reactive oxygen species, which are toxic. They kill pretty much anything. Bacteria, fungi, they will be killed by these reactive oxygen species. Activated neutrophils are actively phagocytic. They'll release their granules, and they will also release these things called nets, which are called neutrophil extracellular traps. Now, this was kind of a totally weird thing. Okay, so this is a bunch of resting neutrophils. As you can see, they're polymorphonuclear neutrophils. So the nucleus is kind of like folded up into this weird shape you can see here. Then when they're activated, you get some of these neutrophils. The nucleus is gone. And what has happened is all the DNA from the nucleus has been expelled from the cells. So you get this kind of like dense network of DNA. And bacteria get caught up in this DNA kind of net, and it's very important for killing of bacteria, apparently. So this is kind of weird. This is like total kamikaze behavior here. And the neutrophil is getting activated, it explodes, releases DNA, and that's actually important for clearing bacteria. Now, there's a link between production of reactive oxygen species and net production. So if you knock out or have an X-linked mutation of the NADPH oxidase, which is essential for ROS production, you don't get any reactive oxygen species and you don't get any nets. And people with this mutation have chronic bacterial and fungal infections, chronic granulomatous disease, recurrent bacterial and fungal infections. So that's telling you that neutrophil activation, this effector branch of the innate immune system, really important, okay, for bacterial and fungal infections. Okay, so what's the important innate response for viruses? The big important thing is the interferon response. So there are 3 types of interferons that will bind to different receptors. So all of the type 1 interferons bind to the interferon alpha R1, R2 complex. Type 2 interferon, interferon gamma, has a different receptor, and the type 3 interferons will bind to a separate receptor. Okay, type 2 interferon gamma, produced by T cells and NK cells. Type 3, produced by epithelial cells, interferon lambda. Also called IL-28A, I think, interferon lambda. Type 1 interferons is more complex. Okay, so interferon beta, this is the one that can be produced by all nucleated cells if they are infected by viruses. Interferon alpha and these other weird ones, epsilon, kappa, and omega, are mostly secreted by plasmacytoid dendritic cells. So PDC are specialists for secreting interferon alpha. So what is these 3 types of interferons have in common is that after the receptors are bind interferon, the signaling pathway will lead to the expression of interferon stimulated genes, ISGs. And there are about 500 ISGs, so 2% of all of our genes, and they have direct antiviral effects on different steps of the virus replication cycle. So the IFI-TM proteins, interferon-induced transmembrane, they inhibit membrane fusion. So they'll inhibit the early steps of viral infection by envelope viruses. Some ISGs inhibit decapsidation, others will be targeting replication or release of virus particles. Other ISGs will actively destroy or degrade virus genomes. So the oligoadenylate synthase RNase L pathway, this will activate an RNase which will destroy all RNAs in the cell. Viral RNase, cellular RNase. ApoBeg will induce a bunch of hypermutation in viral DNA and inactivate the genome because it's been mutated so much it loses all of its sets. Okay, so in terms of signaling, all interferon receptors will induce phosphorylation of either the STAT1 homodimer or STAT1, STAT2 heterodimers. And there are people who've got loss of function mutations in STAT1. So these people cannot or do not have any functional interferon system, either type 1, type 2, or type 3 interferons. So what do these people have? They've got susceptibility to mycobacterial infections, also to other intracellular bacteria, and they can have fatal viral infections. So intracellular bacteria and viruses, that interferon, very important for these. Interferon gamma probably is the most important one here for mycobacterial infections. And type 1 interferons, maybe type 3, probably most important for the viral infections. Now, there are also gain of function mutations in the human population in STAT1, and people with these mutations have got suffer from chronic mucocutaneous candidiasis. So that's telling you that there's some antagonism between interferon, antiviral immunity, and Th17 antifungal immunity. So if you've got chronic activation in your interferon system, that's going to have a negative impact on your resistance to fungi. So another indication of how important a functional interferon system is comes from the study of severe COVID-19 cases in 1920, 1921. So this is in adults. There were mutations in some protein in the interferon system in 3.5% of adult patients with life-threatening COVID-19 compared to patients with mild symptoms. And TLR7 is on the X chromosome. So you've got an X-linked deficiency in about 1% of men less than 60 years old with life-threatening COVID-19. And you also find the same kind of pattern in children with COVID-19 pneumonia. So as you probably remember, generally children have a mild form of COVID-19, but there are some children who have a very, very severe infection. And about 10% of the children in this cohort had mutations somewhere in the interferon, type 1 interferon system. Okay, so this is the underlying susceptibility that this subpopulation had. So another thing that was really kind of brought out with the COVID-19 epidemic was that some people, as we get older, develop autoantibodies against interferon, type 1 interferons. And this was a great study from 2020, mostly from France. So, woo! And if you look at the titer of anti-interferon alpha antibodies here in the severe life-threatening COVID patients, there's a whole bunch of people with these anti-interferon alpha antibodies, almost nothing in people with asymptomatic infection or the healthy controls. And so they actually had these anti-interferon antibodies in about maybe 10 to 15% of patients with critical COVID-19. So the difference here depends on the threshold for positive response. So this is like higher titer. This is like if you lower the bar a little bit, you find that maybe 14% of people have these antibodies. And they also found these antibodies present in almost 18% of people who died. So this is not unimportant here. This is a big effect. If you've got these antibodies, they neutralize the interferon alpha that's released. You don't get a good interferon response. And that can cause you to die. Okay, that's really important. Okay, so we've got about 20 minutes for adaptive responses. I think that's okay now. Okay, so oh no, I'm missing a slide out. No, yeah, okay, yes, that's true. So one last thing before we leave the innate response. So we've seen neutrophils, part of the cellular response, cellular innate response, but there are also innate lymphoid cells in the submucosa that are very, very important. Now, I'm not a big fan about, I'm not a really knowledgeable person about innate lymphoid cells because they didn't exist when I was learning immunology a long time ago. However, what I can read from my slide here is that you have, as far as I understand it, the idea is that, innate lymphoid cells will kind of correspond to the types of T helper cell different subsets that you have. So ILC1s will be activating the same type of effector mechanisms as Th1 T cells. IL2s kind of similar to Th2 T cells, and ILC3s. So these are the ones that are going to be kind of like Th17 T cells. Okay, so that's my understanding of it. So these ILCs, innate lymphoid cells, they apparently don't detect pathogens directly, but they'll be activated by effector molecules that are released either by the epithelial cells in the mucosa or the mononuclear phagocytes like dendritic cells. Well, okay. Broadly speaking, DC, you can consider them as part of the NMPs. So depending on what kind of pathogen the epithelial or the dendritic cells encounter, they'll secrete different types of cytokines. So if dendritic cells here encounter viruses, for example, or intracellular bacteria, they'll secrete IL-12, type 1 interferons, and this will activate ILC1 innate lymphoid cells, which will secrete interferon gamma and activate monocyte macrophages. If helminths are present, these will be detected by the epithelial cells, which will release IL-25, IL-23, and TSLP, which will activate ILC2s. They release IL-5, IL-13. That's going to recruit eosinophils and activate the epithelial cells. ILC3, if we think about fungi and extracellular bacteria, the effector molecules here, the main one is going to be IL-23, or rather the activator molecule IL-23, which will activate ILC3s, and they will secrete IL-17 and IL-22. Okay, so why is this useful? Well, they are directly there at the site of infection, so they can respond very, very quickly with a response that's adapted to the kind of pathogen that's being detected. Okay, so now I'm going to go over to the adaptive response. So when you think about timing, the adaptive response is maybe days, weeks after infection, at least 1 week, I would say. And here, the specificity, how to identify, detect a pathogen, depends on binding of the T cell receptor or the antibody, the BCR, to a specific epitope. So you've got one particular clone which is going to bind to a particular epitope present in a pathogen. So what are the secreted and the cellular aspects? Where you've got secreted proteins are immunoglobulins, antibodies, and the cells. We're talking about T cells and B cells. The big important thing about the adaptive response, well, two things, I guess: high specificity, very, very specific for a particular pathogen, and immunological memory, because these memory T cell and B cell clones can persist for years, maybe decades. Now, what is essential is the link between the innate and the adaptive response, and that's really the important role of dendritic cells, which are going to be covered or express a lot of different PRRs. They're present at the mucosal surface. When they're activated, they migrate to the draining lymph node, and that's where they can interact with T cells. Okay, so depending on the type of dendritic cell and the type of pathogen that's been encountered, you'll get different types of activator cytokines secreted. So conventional DC1s that are express this molecule XCR1 will mostly be detecting viruses, maybe bacteria and protozoa. They'll secrete IL-12 and activate Th1 type immunity. Conventional DC2 will be detecting helminths, fungi, and if fungi and extracellular bacteria activate conventional DC2, these cells will secrete IL-1 beta, IL-6, IL-23. That's going to give you Th17 response. CDC2 that activate a Th2 response, not really clear if they secrete some specific molecule which will be directing differentiation of T cells to Th2. So it's more like the overall environment with IL-4 being secreted by other types of cells. Okay, so the result here is that the type of adaptive response you get will be different according to the type of pathogen. So if you've got a Th1 response, typically you're going to have CTL and IgG. Th2 response, typically IgE and eosinophils. Th17 response, IgG, not really many CTL. Okay, so we got about 15 minutes to clear up T cells, B cells, antibodies, and everything. So CD4 T cells, as you know, recognize antigens presented on HLA class 2. Their role is to orchestrate and orient the adaptive response. So they're going to be activated in the draining lymph node, and they differentiate into effector cells that then remigrate to the site of infection. And they'll recognize peptides that are presented on HLA class 2, mostly, I guess, on mononuclear phagocytes. Most somatic cells will not express HLA class 2. Depending on their differentiation, they'll have different functions. So Th1, differentiated T cells, their big effector molecule is interferon gamma, so that will activate the mononuclear phagocytes and give them the ability to kill intracellular pathogens. Th17 T cells, their big favorite cytokine is IL-17, which will activate neutrophils. Th2 effector T cells secrete IL-4, IL-5, activate eosinophils. So if you've got no CD4 T cells, what happens? Well, that's what happens when you've got AIDS, right? You've got HIV infecting, killing all your CD4 T cells. If you've got less than 50 circulating CD4 T cells in your body, you're going to start getting multiple opportunistic infections. Pneumonia, tuberculosis, candidiasis, toxoplasma, and some DNA viral infections, also some kind of cancers. So this is telling you that CD4 T cells are playing a central role in the immunity to all of these different types of pathogens, okay? What about Th1, Th17, for example? So if you have a defect in your IL-12 interferon gamma pathway, different mutations that people can have, then you'll have susceptibility to mycobacteria and salmonella. So once again, this is telling you, just like the STAT1 deficiency, that interferon gamma really essential for immunity to intracellular bacteria. And if you have a loss of function mutation in the IL-17 receptor, then you get Candida and Staphylococcus aureus dermatitis. So this Th17 immunity, fungi, and extracellular bacterial pathogens. Okay, CD8 T cells. Well, they recognize peptides on the surface of class 1 and essential or important for responding to intracellular pathogens, viruses, intracellular bacteria, intracellular parasites. So when they're activated, they secrete interferon gamma and they kill infected cells. Now, there are some specific deficiencies in CD8 T cells, which are related to either the lack of CD8 or the lack of presentation on HLA class 1. And people who've got these deficiencies, they show lung inflammation, don't know why that happens, and many opportunistic bacterial infections. Surprisingly, no history of severe viral infections. So maybe, well, what I understand by this is for an acute viral infection, maybe it's the interferon response, the innate response, which is the most important. Okay, CD8 T cells determines whether you can eliminate the path of the virus or not. And you can also see this in associations between HLA polymorphism and disease outcome in viral infections. So HLA class 1 is associated with HIV progression. HLA1 and HLA class 2 associated with the elimination or not of hepatitis C virus. HLA class 1 polymorphism also involved in the severity of malaria. SARS-CoV-2, HLA class 1, not really very clear. Okay, so let's have a look at the difference here between HIV and SARS-CoV-2. HIV, you've got a chronic infection. CTL are going to be this association telling you that CTL are important in, controlling the infected cells. SARS-CoV-2, let's have a look. Okay, so HLA genotype associated with HIV disease progression. So this was a big paper in 2010. They did a GWAS study, one of the first big ones, almost 1,000 HIV controllers, 2,600 HIV progressors, and they're looking for the differential presence of some genotypes in the controllers. And they only found this thing here, one massive peak. This is HLA class 1. So that means what? That means that the CTL response is the main driver for this phenotype, whether you can control the replication of HIV or not. And this is in patients who were not on antiretroviral therapy. Now, what about SARS-CoV-2? So this was 2021, big consortium. Once again, more than 1,000 cases, 1,000 controls. HLA class 1, nothing, no HLA signal. What you do get are some of these molecules, interferon alpha receptor, OAS1, interferon. Okay, so interferon here is what's critical, not CTL, not T cells. Acute infection, whether you get severe infection, live or die, it's interferon. It's not T cells. Okay, so what's the role of CTL in an infection? So it's not so easy to get data on primary infection in humans because you don't really know the date of the infection a lot of the time. So this is why animal models were very important here in mouse and in non-human primates. So I'm going back to some work that was done, 25 years ago now, maybe more, in SIV-infected macaques. So what you can do is you follow the viral load and you follow the presence of CTL. And what you can see is when the CTL go up, the virus load goes down. Okay? And that's a reproducible finding. You get CTL detectable 2 to 4 weeks post-infection, and when you can detect CTL, then you can see the viral load goes down. That's a correlation, not causality. So to prove causality, you have to perform an experiment. So what they did here was they depleted CD8 T cells before infection with monoclonal antibodies. And if you do that, well, you get some monkeys that have partial CD8 depletion. These are in the controls. You've got the peak of virus replication, then it goes down. If you've got animals that have persistent CD8 depletion, you've got the peak of viral load and it stays really, really high. Okay, so in this infection, SIV, and I think it's a pretty good model for HIV, your CTL response is coming in, 2 to 4 weeks post-infection. The role is getting rid of the infected cells, clearing the infection. Okay, and if you don't have a CD8 T cell response in this model, the animals that were CD8 depleted, for more than 4 weeks, they all died in less than 6 months. The animals with a normal CD8 response, the control, of 3/4 of them survived for a year. So that's the role of CTL. At the end of an infection or in a chronic viral infection, eliminating all the infected cells. Okay, 5, 6, 7 minutes on antibodies. So 2 types of antibody responses to pathogens, T cell dependent, and here you get isotype switched high affinity antibodies. And the type of antigens that are involved in T dependent responses are individual pathogen proteins or glycoproteins. So you can have multiple epitopes on one single protein. So typically that will be bacterial exotoxins or a virus envelope glycoprotein. And after you have this differentiation of B cells into long-lived plasma cells, they can continue to secrete antibody for years. Okay, this is your serological immune memory. And that's what's going to protect you from future infections, especially secreted IgA. Now, as well as that, some pathogens induce T-independent responses. So this is mainly IgM, low affinity, no affinity maturation, no class switching. And these IgM memory B cells, generally short-lived. And the type of antigen that induces an T-independent response is when you've got a lot of repeated epitopes on the same pathogen surface. So this can be bacterial capsid antigens, Plasmodium falciparum merozoite antigens, probably trypanosome surface glycoproteins, and I think non-enveloped virus capsids have definitely got something like that as well. When we publish it, it's going to be so great. I'm telling you, this is going to be fantastic. Okay, so when does the antibody response occur? So we got some pretty good data from SARS-CoV-2 here. Very good for revisiting classical immunology. So we've got, this is the time post the onset of symptoms, okay? And this is in black IgM and in the clear symbols here IgG against the virus. So you've got seroconversion, detection of antibodies, 2 to 3 weeks post symptom onset. So probably a little bit delayed compared to CTL. Often you have CTL first, then you get antibodies. And probably IgM a little bit earlier than IgG. But it's not really a long time. It's maybe just 2 or 3 days. And then you get this classical thing where afterwards IgM goes down and IgG is maintained. So if you look a little bit longer, you've got these IgG memory B cells. You know, they stay at a high level for the first 6 months. IgM specific B cells, where they start going down, can't detect them after 6 months anymore. Now, a little bit about class switching. So the whole point of class switching is for, well, antibodies to gain different effector functions. Okay, so IgG, some isotypes can activate complement. Some of them can be recognized by Fc gamma receptors on different types of cells. IgA, it's secreted at mucosa and can also be recognized by Fc alpha receptors. IgE, recognized by Fc epsilon receptors on eosinophils and mastocytes. So the isotype will determine what kind of effector function is associated with the antibody. Okay, and so how do antibodies inactivate or kill pathogens? Okay, so there's different mechanisms involved. So one of them is opsonization. The antibody is going to bind to the surface of the pathogen, and then it can induce phagocytosis by mononuclear phagocytes or neutrophils via the Fc receptors or by the deposition of complement. So this is very important for immunity against. So let me just go through my little symbols here. This is gram-negative bacteria. This is gram-positive bacteria. This is protozoa. This is fungi. And this is yeast. Okay, so that's what my icons mean here. So all of these pathogens will be susceptible to this effect, opsonization, and then phagocytes come and destroy them. Lysis, okay? So if antibodies induce complement deposition on the surface of the pathogen, that can kill gram-negative bacteria and protozoa. They're sensitive to complement-dependent lysis. Another mechanism is just neutralization. The antibody binds to something on the surface of the pathogen, and then it won't be able to infect you anymore. So that's typically what we think about for viruses, but it's also true for gram-negative, gram-positive bacteria and protozoa. So all of these types of pathogens often will need to express an adhesin that allows them to bind to a particular host cell. And if you can block that, then they won't be able to infect you anymore. And final is a mechanism to inhibit infection is when antibodies inactivate bacterial exotoxins. So the bacteria will be able to infect us, but they won't be able to kill us because the exotoxins that are related to the pathology are inactivated. So that's a mechanism that's important for immunity to gram-negative and gram-positive bacteria. So different types of antimicrobial vaccines work by inducing antibodies against the pathogens, and the effector mechanism is different depending on what the pathogen is. So pneumococcal vaccines, these are the capsule antigens. Really, their big effect is in so against pneumococcal. So that's Streptococcus pneumoniae, gram-positive, and they work because they allow the opsonization and the destruction of these bacteria. Meningococcal vaccines, Neisseria meningitidis, it's a gram-negative pathogen. These antibodies can induce the lysis of the bacteria by complement. Neutralization, all antiviral vaccines that actually work are based on the induction of antibodies that will neutralize the virus. The malarial antisporozoite vaccine, which I believe has been licensed, been developed by the OMS, is recommended now. It's based on inducing antibodies against the circumsporozoite protein that will prevent the infection of hepatocytes. And pertussis vaccine also works this way. And toxin inactivation, this is diphtheria, tetanus vaccines. They're based on the toxoid, the formaldehyde inactivated exotoxin. Okay, so that's about it. Just about on time here. So this is the big summary table, which I think I will not read out for you, but that's just everything about anti-pathogen immunity on one slide. One thing I will say is, okay, innate response, this is the one determines whether you're going to live or die. CTL determines how quickly or whether you can eliminate the infection. Antibodies, CD4 T cells, this is your immune memory, whether you are immune to subsequent infections. They arrive just kind of at the end of the infectious process. It's kind of cleanup and immune memory. Okay, so these are the different phases of the anti-pathogen immune response and their importance, wow, for different aspects of immunity. Okay, thanks everyone for listening, and I'm amazed I did okay on keeping the time this year. Last year was very, very bad.